right, so good morning. So today we're going to go over finishing up chapter 3. Just wanted to make sure you remember that we're in week 4. So in week 4, we had Labor Day yesterday, so we're finishing up chapter 3, cellular level of organization. And Thursday on Honor Lock, you will have your exam 1 over chapters 1, 2, and 3. Make sure to go over the exam 1 review sheet. I believe it's in week 1, so you know what to study for, and then go over your full notes in your lecture presentations. Make sure you're ready for those questions. All right. So I believe we left off at microvilli last time, so we'll pick up here. So we were going over parts of the cell. So microvilli. So these are small projections, particularly in comparison to cilia on the cell membrane, and the purpose of them is to increase your surface area along your cell membrane so you have more exposure to the extracellular environment. So the cells that are actively absorbing materials, like your cells in your digestive tract, right, need to be able to absorb your food. So those membranes are covered with microvilli. So those are the microvilli. You also have centrioles. And these are a pair of rod-like structures. You usually see them near the nucleus. And inside each centriole, you're going to have 9 bundles of microtubules, and each bundle is going to have 3 microtubules in it. And what do? Well, they help form the mitotic spindle that helps pull your chromosomes apart during cell division. Also, they help in organizing your cytoskeleton. Another term, the centriosome, is basically the cytoplasm surrounding those centrioles. So you basically put the centrioles together and you get a centriosome. Now the cilia, they're hair-like projections. And they are shorter than flagella, which we'll talk about later. All right, and their arrangement is you have basically 9 groups of 2 pairs of microtubules. And these are found on cells. You see them in some lower animals. You also see them on some specialized cells in higher animals. Okay, so like some bacteria you might see them on, but you also see in certain cells in humans, like the respiratory tract. And that's to help move things away from those cells. All right, so where do you find them in humans? We mentioned the respiratory tract. You also find them in the female genital tract as well. So how are they anchored to the cells? They have a basal body that anchors them. And how do they move? Well, they beat rhythmically, okay? And they help to move fluids or secretions across the cell surface. And the flagella are the longer projections, and they move kind of like whips. All right. So they also have an arrangement on the inside that is basically 9 pairs of microtubules and 2 pairs in the middle. So again, you'll see them on some lower animals. You'll also see them on some higher animals in specialized cells. So in the human body, sperm cells are the only cells that we have, or that humans would have, that have flagella, right? And those are in the male reproductive system. Their role is to help move that cell through some surrounding fluid. Okay, so cilia are going to be the ones that move the fluid past a stationary cell. Okay, flagella are involved in moving the actual cell. All right, next organelle we're going to look at is the ribosome. It's intracellular, which means it exists inside cells, and its job is to make protein. Okay, this is where protein synthesis happens. So it's made up of light and heavy ribosomal subunits, and they contain special proteins as well as ribosomal RNA. And ribosomal RNA is made inside the nucleolus. Remember, it leaves, maybe we haven't covered it yet, but it's made in the nucleolus, leaves the nucleus through the nuclear pore, and is able to make proteins in the cytoplasm. So where do you see them? You see them as free proteins floating in the cytoplasm. You also see them as fixed ribosomes that are going to be attached to the endoplasmic reticulum. Okay, when they're attached to the endoplasmic reticulum, that endoplasmic reticulum is considered rough, or rough ER. So let's talk now about organelles that have membranes. So first we're going to cover the mitochondria, known affectionately as the powerhouse of the cell, responsible for the overwhelming majority of the ATP that's made in a cell. Remember, ATP is our cellular energy source. It has a double membrane, okay, so 2 layers, and it has folds on the inside, and those folds are called cristae. So an individual fold would be crista, plural, all the folds are cristae. So inside the mitochondria, you also have fluid, and that fluid is called matrix. Believe it or not, you actually have some DNA inside the mitochondria, okay? And so it's going to have a separate form of RNA from what you have that's made by your DNA in your nucleus, okay? So it has its own DNA and RNA. And it is capable of dividing on its own, okay? So when the cells divide, you also have to divide the organelles, okay? So these organelles can divide, so that you have enough organelles to go to the 2 new cells. So there's a lot of theories on mitochondria and why we have them. So the current prevailing theory or idea is that mitochondria is basically a remnant of a bacterial invader that entered a human cell in some sort of symbiotic relationship form, so they stayed. They got to live inside the cells, and they got to make ATP for the cell. So both cells benefited. All right, but now we consider it to be an organelle. All right, so let's shift. Now we're still talking about the mitochondria, so let's talk a little bit more about what happens inside of it. So here we're sort of shifting from structure to function. So here we're doing a little physiology. So one of the processes you need to know that happens inside these cells is glycolysis. And glycolysis, basically, glyco is a root word that means what? Anybody know? Sugar, right? It looks kind of like glucose. Lysis is a root word that means to do what? Break. So we're breaking sugar. So we're a 6-carbon glucose molecule is broken down to a 3-carbon molecule of pyruvic acid. So if you take a 6-carbon compound, you break it into a 3-carbon molecule, you basically broke it in half, right? So you made 2 molecules of pyruvic acid. And that mitochondria then will take away a carbon dioxide molecule from each pyruvic acid. Okay. Now, the next process that you need to know is the tricarboxylic acid cycle, or TCA cycle. You might also see this as the Krebs cycle somewhere, Krebs. That's named after one of the scientists who studied it. But they all mean the same thing. You may also see citric acid cycle. All right. It's an enzymatic pathway, which means, again, it uses enzymes, which are proteins, right? And they lower the activation energy of chemical reactions. And they're going to be breaking down organic molecules. And remember, we learned organic molecules have carbon, hydrogen, and oxygen in them, right? So then they have electrons. Remember, we learned about that in chapter 2. These are negatively charged subatomic particles that are removed. And what do? They convert ADP to ATP. So they convert adenosine diphosphate to adenosine triphosphate. Remember, we learned about those molecules at the end of chapter 2 in our discussion of nucleic acids. So aerobic metabolism. So aerobic means requires oxygen. So air in aerobic, think of it as air, which, you know, for us, what we really need out of the air is oxygen. Okay? So, like if you do aerobics or aerobic exercise, you're doing exercise that's going to require a lot of breathing, right? So, it requires oxygen. Here you're breaking down organic substrates, those are reactants, into carbon dioxide and water, which are products, via that pyruvic acid that we just made in glycolysis. Okay, so those processes are very important to the mitochondria, and that's how they make ATP. That's how they become the powerhouse of the cell. Next, we're going to talk about the endoplasmic reticulum, another membranous organelle. It's basically just a network of membranes inside your cells, and they connect the nuclear envelope, or connect those membranes that connect to your nuclear envelope. And it should have an E at the end of, envelope, it should be envelope, or your nuclear membrane. So the ER is going to form what we call hollow tubes, flattened sheets, and another thing called cisternae, which are round chambers. And cisterns, historically were chambers, so we're using that in the science context, a cisternae. So what are the functions of the ER? Well, they are synthesis, which means they're making things like lipids, carbohydrates, and proteins. They store things, so they contain molecules or different materials that are absorbed in your cytosol. Remember, that's the fluid in your cytoplasm. They're also involved in transport, which means they can make material go from one place to another place. And we'll learn about how they do that. They're also involved in detoxifying substances like drugs and other toxins. And they have enzymes inside the endoplasmic reticulum that can serve to neutralize those chemicals. So there's two types of endoplasmic reticulum. You have the rough ER and the smooth ER. Let's cover the smooth ER first. So the smooth ER is smooth because there are no ribosomes on the surface. Because there are no ribosomes, that means it can't make protein. So what does it do? Well, I'm glad you asked. Well, instead of making proteins, it makes lipids and carbohydrates. All right. It also stores calcium in the case of striated muscle cells. Okay, so that's the smooth endoplasmic reticulum. What about the rough endoplasmic reticulum? Well, it's going to have ribosomes on it, and that's what makes it rough. So it does get to be involved in protein synthesis. It does make proteins. It also will modify those proteins chemically and package them and send them on to the Golgi apparatus. Okay, all right, so let's go ahead and take a break, and I want you to explain to your partner the difference between rough ER and smooth ER and what they do. So, and if you're at home, you can kind of practice doing this on your own. All right, let's talk about the Golgi apparatus. So with the Golgi apparatus, you have these flattened membrane discs, which we call saccules. And what does the Golgi apparatus do? Well, it's involved in packaging and synthesizing, or making and packaging secretions. Some examples of these secretions, things you secrete or release, could be hormones. They could also be enzymes. Okay, they could also be involved in renewing and modifying the cell membrane. We'll talk about how that works in just a minute. They're also involved in packaging certain enzymes within vesicles for use inside the cell. Okay, and we'll talk about what vesicles are. But basically, vesicles are spheres. So they're kind of like little balloons inside your cell that transport stuff inside of them. And stuff is a fancy anatomical term, right? So just using that because you can transport different things. So they can these transfer vesicles can move certain materials from one saccule to another. So you have 3 different kinds of vesicles that you make at the Golgi apparatus. Some are secretory, which means they have secretions, and they're going to be released from the cell through a process called exocytosis. Again, don't get bent out of shape over these words. What's exo mean? Outside. What's cyto mean? Cell. So it's the process of moving things out of the cell. All right, so you also have process of making new membrane components that exist on the cell membrane. So the Golgi apparatus can actually change the properties of a cell membrane over time. Okay, packaging intracellular enzymes. Okay, we'll learn about lysosomes next, and they have digestive enzymes in them. So the lysosomes are made by the Golgi apparatus. Again, have these digestive enzymes in them. They can be primary or secondary. If they're primary, that means their enzymes are inactive. And if they're secondary lysosomes, your lysosomes are going to be active. Okay, so let's make sure we keep our masks over our mouth and our nose. Okay, make sure, I know they're easy to come off, so make sure you keep them on. All right. So just remember the secondary lysosomes are actually the ones that have the active enzymes. And what do? Well, they will fuse or merge with vesicles to release the enzymes, and they cause digestion or breaking down of whatever's in those vesicles. That can also fuse with worn out cell organelles to digest them. That's called autophagy, or self-eating. Because remember, auto means self. Like an autobiography is a book you write about yourself. Phagy means eating. So, small point here, organelles have a certain shelf life. Okay, so they don't last forever. Sometimes they get worn out and they have to be degraded. And the process and the parts can be reused. All right. Lysosomes can also help you with immunity. They can break down bacteria that are brought into your cell via endocytosis. So if exocytosis is bringing things out of your cell, what is endocytosis? Bringing things inside the cell. Endo for inside. Generally, lysosomes are involved in cleanup and recycling. They're also sometimes called suicide sacs, and they can basically, if they break their cell membrane, or not their cell membranes, if they break the membrane of the lysosome, those digestive enzymes would actually destroy the cell, and that's called autolysis. Remember, auto cell, lysis is breakdown, and this is actually a normal process. You do have some cells that you want to do this. Okay, so your body sort of has a self-destruct mechanism, and it's not going to do this for all your cells, but it can know sometimes this cell's beyond repair, or this cell's got something in it that we don't want to, divide and reproduce more of itself. And so it has a process called programmed cell death. All right, so that's the lysosomes, not to be confused with the peroxisomes. And these have enzymes in them that absorb and neutralize toxins like alcohol and hydrogen peroxide, hence peroxisomes. And their role is to protect your body from free radicals. And free radicals cause cascades of reactions that basically make cells unstable and parts, chemicals unstable. So basically all your organelles that have membranes are either connected or communicating with each other based on movements of vesicles. So vesicles are kind of like, text messages, right? So they can be sending each other messages, all right? So membrane flow, another concept. It just means that there's continuous exchange and movement of these vesicles. All right, and they form the cell membrane, and they can remove and recycle segments of the cell membrane. So before we go on to the nucleus, I want to make sure you understand vesicles, because they have a membrane around them. When we talked about them changing the composition of the cell membrane, if that's the goal, when they attach to the membrane, they sort of just become part of the membrane. So they could maybe put new membrane proteins or carbohydrates or some other material on the surface of that membrane. Okay, so maybe you need more receptors for something, and so that's one way you can get those receptors. More channel proteins. All right, so let's go on to the nucleus. It's your control center. Okay, it's where kind of all your direction happens. Determines your structural and functional characteristics, and it does that by controlling which proteins are going to be made and how much they're going to be made. So your nucleus has a nuclear membrane or nuclear envelope, and it surrounds your nucleus, and it's a two-layer membrane. Has what we call a perinuclear space. Your nucleus also has holes in it, or pores. Allows for communication of chemicals between the nucleus and the cytosol. It's also where the ribosomal RNA leaves the nucleus. So just like you have cytoplasm inside your cell, you have nucleoplasm or fluid inside your nucleus. And just like you have sort of a cytoskeleton in your cell, You have a nuclear matrix inside your nucleus. Here's your nucleus, some structure, has some filaments in there. Now, inside your nucleus, you're also going to find DNA. And most of the time, your DNA is going to exist in what we call chromatin. Okay? Because most of the time, the cell's not dividing. There's only a small amount of time where the cell's actually divided. And it's basically a tangle of different filaments. All right, so when your cell's getting ready to divide, your cell starts forming nucleosomes and chromosomes. So your nucleosomes is basically a complex of DNA strands start winding around these proteins called histones. Okay, and then eventually you start getting enough of those, you get chromosomes. Now, inside your nucleus, you also have an area called the nucleolus. Plural is nucleoli, because some cells have more than one. And it's basically an organelle inside your nucleus, and its role is to make ribosomal RNA, or ribosomes. And we learned about RNA in chapter 2. Remember, it's a nucleic acid. Sugar is ribose. It's ribonucleic acid. All right. As we said, the nucleus is involved in DNA and directing the activities of the cell with that DNA. Well, how does it do that? It does that through what we call the genetic code. So here we got two terms, or really three terms you really need to know well. So you might put your stars by it if you have a printed copy, or highlights or whatever you want. But I'm basically making sure you know these. So your overall method of storing information inside your DNA and your nucleus is called a genetic code. And specifically, it's called a triplet code because each amino acid, remember that, is a building block or monomer of protein, is identified by a sequence of 3 nitrogenous bases, which remember is a component of a nucleotide. And a gene basically has all the triplets that you need to make a specific protein or polypeptide chain. And every gene is going to have different segments that regulate its own activity. So that these segments can regulate what happens to that gene itself. And in a related process is one called DNA fingerprinting. All right, so in your cells, or in your DNA, in your cells, you actually have some parts or strands of DNA that are basically repeats. And the number of repeats is considered a fingerprint. It's basically a way of identifying you as an individual and also the characteristics of you. All right, let's see, page 22. There's a lot of really important information on this page. So, I mean, obviously, everything is important. We try to summarize it, a whole big chapter into 30 pages. Page 22 has got a lot of good stuff on it. So make sure you spend a lot of time on this page. All right, so gene activation and protein synthesis. So before your gene gets activated and you start protein synthesis, you start making protein, you need an enzyme called RNA polymerase to bind to that gene. And remember we said ASE at the end of any word means it's an enzyme. Okay, and it's basically, the name of it is kind of telling you what it does in most cases. So this enzyme's job is to make an RNA polymer. And remember, a polymer is basically a link of monomers. And so how do we make proteins? Well, there's two steps, two big steps anyway, or big processes, which each have their own set of steps. First process is transcription, second process is translation. Make sure you know the difference between these two. Transcription is the process of transcribing DNA or decoding DNA. Translation is the process of making protein. More specifically, in transcription, DNA is used to make RNA. In translation, RNA is used to make protein. Okay, one thing I want to make sure you understand that is not the case. DNA does not become RNA. RNA does not become protein. They are different molecules. They are used as templates to make the next piece. So DNA is used as a template to make RNA. RNA is used as a template to make protein. But that whole thing, DNA to RNA to protein, that's a huge concept you need to make sure you have imprinted in your brain. Okay? I can guarantee you're going to have several questions that basically ask you that in different ways to make sure you understand it. Transcription, you use DNA to make RNA. Translation, you use RNA to make protein. Okay, so let's focus on transcription. And again, here our role is to make RNA. And there's three different kinds of RNA, or ribonucleic acid. You have the messenger RNA, or mRNA for short. It's just your linear shape or sequence. And it will attach to the ribosome in the cytoplasm. So it's going to have to leave the nucleus and meet up with the ribosome in the cytoplasm. Next type of RNA is transfer RNA, or tRNA for short. Has kind of a clover leaf shape, like a 3-leaf clover or 4-leaf clover. And its job is to pick up an amino acid in the cytoplasm and essentially bring it to the ribosomal RNA, which is the third category, or rRNA. Its job is to help make up the ribosomes. Typically, they're going to have two subunits of rRNA. You have a big subunit and a small subunit. Another concept you definitely need to understand is the difference between a coding strand and a template strand. So your coding strand, remember your DNA has two strands, is double-stranded. So your coding strand is the strand of DNA that has the triplets that will specify your sequence of amino acids. Your template strand is the other strand of DNA and it has the complementary triplets that we use as a template for mRNA production. So the mRNA is going to have a nucleotide sequence that's identical to that of the coding strand, with the exception that RNA, remember, has uracil as a nucleotide and not thymine. So both strands are used, but both strands are not copied. All right, so let's see. Just want to quickly make sure you understand the difference. Let's see if I can pull up. Office. about that, y'all? So, all right. All right. We have ACG and T. All right. So that's our coding strand of DNA. Let's make this larger so everybody make sure you can see it. So our template strand is complementary. So our template strand, T is complementary to A. What's complementary to A? U for, it'll be U for DNA, but I mean U for RNA, but we're talking about DNA right now, so it'll be T. C is complementary to G. All right. So that's DNA. Now, for making messenger RNA and transcription, so it's going to basically be the same as the coding strand, with the exception of there's no thymine. So it's, all right. So the mRNA looks like the coding strand, except all the T's are replaced with U's. Okay. And that's just going to take practice. So if that's not quite making sense just yet, don't worry about it. Okay. But just wanted to show you that so you at least had something to start thinking that through. Yes. On the second one, on the mRNA? All right, so like right here? Thank you. So I have that one backwards, okay? Thank you. All right. Yep. Thank you. I need to drink my coffee. That one didn't change, right? Because there's no U's or any A's in there. So, all right. So I'll give you a minute to rewrite that last strand, or mRNA strand. Keep me honest. All right. I'm okay with that. All right, so let's go back. Okay. So other terms you need to know are codons. You have a nitrogenous base sequence on the mRNA. And it's going to match an anticodon on your transfer RNA. So the mRNA has codons. The anticodon is going to be on your transfer RNA. And again, it's going to match the codon on your mRNA. So I'm going to try this one more time, make sure it doesn't click on other stuff. So let's go back to this, let you practice. So this time, what you do. Template strand and mRNA strand. All right. So let's see. Okay, Jalen, what's going to be my first DNA codon here? My first one. So what would be our corresponding nitrogenous base for A? T. What about for T? All right, and for G. Thank you. All right. Let's see, Johanna. Okay, so what's going to be my next one? EGC. Okay. Let's see, Aaliyah. What's the next one? GGT. Great, great, great. Naya. What's the next one? Thank you. Very good. All right. Let's go to the mRNA strand for transcription. All right. Let's see. Danielle. AUG. Okay. Let's see. McKenna. Okay. Robert. Correct. And let's see. Ashton. I can't hear you. Yes. Okay, so for A, what's the complementary base in RNA? A binds with, well, T in DNA, but what will it bind in RNA? U. All right, and for T will bind with A, and then A again will bind with U. Okay. So again, no problem, just our practice, all right? But this is just to give you some opportunity to practice. All right. So it's okay to not know today. It's okay. I want you to make sure you know on the exam, okay? All right, so just go back over this. You can watch the video again. I'll post it later today. All right, so let's see. Let's get into transcription. So again, this is the process of taking a DNA template and making RNA. So after your mRNA transcription, your messenger RNA is going to take the instructions from the nucleus out to the cytoplasm. So in step one, your DNA is going to unwind. Okay, because remember, it's wound up in a double helix. So the strands are going to come apart. The enzyme RNA polymerase is going to attach to the template strand. And your nucleotides in the next step are going to attach to your template strand DNA according to base pairing rules. Okay? All right, so if you have ATC is a nucleotide in your DNA, corresponding base pair in the RNA is UAG. Next, you're going to see a stop signal. So it's going to tell you the process to stop transcribing. Your next step is RNA processing. So you're going to have editing of the mRNA strand. So the introns are what we call nonsense regions, parts that you need to take out, they're cut out. And then the exons, the remaining segments, are going to be spliced together. And if you remember, we said that mRNA, once the mRNA is made, does it stay in the nucleus or where does it go? It goes out to the cytoplasm, right? It has to go find a ribosome. So exons, if you want to think about it that way, because exons need to go out, those are the pieces that you need. The introns are the pieces you cut out and they stay behind. Step 6, your mRNA is going to leave the nucleus via the nuclear pore. So mRNA, once it's been edited, can go through the nuclear pore. Go find a ribosome. So this was one of your potential essay questions. Okay, so you want to make sure you know the steps. All right. So make sure you can explain what's going on here. All right, so let's go ahead and pause. I'm gonna let you explain to your partner steps of transcription. All right. So let's talk about translation now. So this is a process where mRNA strands used to make protein or polypeptides. So during translation, you're going to make what we call a functional polypeptide using the information in that mRNA sequence. All right, so the sequence of the mRNA strand, codons, or sequence of codons in the mRNA will determine the sequence of amino acids in your polypeptide. And there's three main parts of translation. You have initiation, elongation, and termination. So you got to start, make the polypeptide longer, then you finish. So in initiation, your mRNA is going to attach to your ribosomes. Then your transfer RNA is going to bring the corresponding amino acid over to the mRNA. So your anticodon and tRNA is attaching to your mRNA codon. Next phase is elongation. You have another transfer RNA. It's going to bring over another amino acid. All right, so it's red rover, bring another amino acid over. Okay, so you're going to have that anticodon bind to the next codon on the mRNA strand. Okay, so you have two binding sites filled. Now you have two amino acids sitting next to each other because the two tRNAs are there. Because remember, the tRNAs have the amino acids on them. Those tRNAs say, I think I'd rather be bonded to you than being bonded to the tRNA. So a peptide bond forms between the first two amino acids. Then the first tRNA can leave. Then you're going to get another tRNA. It's going to bring another amino acid. Then they're going to get another peptide bond, and then that second tRNA can leave. And that happens over and over and over again till you get to the termination phase. Then you get step 6, and it'll continue until you get your stop codon. And at that point, your ribosomal subunits or your rRNAs are going to detach from each other. Now, in some cases, you'll see what we call a polyribosome. Poly means many, right? So here you'll see many ribosomes attached to the same mRNA strand, okay? So that could be a strand where you need to make lots and lots of protein, so it may have lots of ribosomal subunits attached to it. Okay, so that's translation. Taking mRNA and making protein. Okay, so I'm going to give you all a minute. You can explain translation to your partner and make sure you understand the difference between translation and transcription. Okay. So you learn a little bit about production of RNA, production of protein. DNA, again, controls your cell by directing protein synthesis, making proteins. Your DNA is going to have direct control over making structural proteins. Proteins exist in your cell membrane, and proteins exist in your cytoskeleton. DNA is going to have more indirect control over the other parts of cell metabolism. And it does that by regulating the synthesis of different enzymes, which are proteins. Okay, so those proteins then, or those enzymes, will regulate other parts of cell metabolism, but the DNA is going to control production of those enzymes. So let's transition a little bit from the cell, making RNA and protein to the cell reproducing itself in the cell life cycle. So in the cell cycle, it's a period beginning from one cell division to the next one. And cell division is sort of that phase of the cell cycle where one cell is dividing into two daughter cells. So the cell cycle is actually larger than just cell division. And why do cells divide? Glad you asked. Well, they increase the size of the organism, and they can replace cells that are dead or worn out. Remember, we talked about DNA. We'll talk about it again in this context. It's your genetic material found in living organisms, your blueprint. DNA can be found in chromosomes. Okay, these are the structures inside your nucleus that have the genes. And you say for genetic expression. When you say expressing, basically means like making proteins. That's how genes express themselves. They make proteins. All right. Humans are going to have 46 chromosomes. Other organisms could have more or less. The individual genes are individual units of genetic material, or DNA, found on a chromosome. And that's what determines your different hereditary traits, your traits that you inherit from your biological parents. Now, chromatids are basically a copy of a chromosome. And around that chromatid, what we call a kinetochore. It's a complex of proteins surrounding it. Another term, you've heard this already, the centromere. It's a zone of your chromosome where the spindle fibers attach. And that helps to move the chromosomes when it's time to divide the cell. Another cellular process is mitosis. Here you're dividing essentially the nuclei. And your two new cells have the same genetic material as the original nucleus. This happens in somatic cells. Remember the non-sex cells? It's a way to preserve your DNA from generation to generation. Separate process is meiosis or meiosis. And this is the process that the sex cells go through. It's a special type of nuclear division. You reduce the chromosome number by 50%. So you don't end up with the same amount of chromosomes, you end up with half. Or same amount of DNA, you end up with half. And that's because in mitosis, you have one cell division. In meiosis, you have two. All right, you also have cytokinesis, which means you're dividing the cytoplasm. So mitosis, you're really splitting up the DNA. Cytokinesis, you're splitting up the cytoplasm. And that's a part of the phase telophase. So in lab, you should have learned about prophase, metaphase, anaphase, and telophase. Okay? This is part of it. Cytokinesis is part of telophase. All right, a little more information on that. Interphase is where cells spend most of their time. You'll have G0, G1, S, and G2 in interphase. In G0, or the zero gap phase, cells is not preparing for cell division. It's just doing its normal job. When it gets to G1, cell starts making the components of the cytoplasm. It's like your cell membrane, your different organelles. Copying those so you have enough for two cells. And in S phase, S stands for synthesis. That's where DNA replication happens. So replication means to copy. So there you're making a copy of the DNA. And here each daughter cell is going to have another complete set of DNA. So you go from one set of DNA to two. So you have enough for each daughter cell. Then you go to the second gap phase, and that's where protein synthesis happens. Okay, because you're also going to need proteins for each cell. And that's separate from really the protein synthesis that you have just to sustain that cell. This is to make enough for two cells. Then you get to M phase, or the mitotic phase, where mitosis or meiosis occurs. Then in prophase, you start getting DNA condensing. The DNA starts condensing and making chromosomes. The nuclear membrane starts to disappear. It's harder to see. Okay, you start having the spindle fibers attached to the chromosomes. Okay, so where the centriole starts showing, you start seeing chromatin start condensing in the chromosomes. In metaphase, look at the M for metaphase as the middle, right? Chromosomes align in the middle are the metaphase plates. So if you see that, it's metaphase. In anaphase, then the sister chromatids in the chromosomes split apart. Start moving to opposite sides of the cell. In your telophase, chromosomes are going to be at opposite sides of the cell or opposite poles. You start seeing more cytoplasm divided. Big thing there is you start seeing the cleavage furrow. Okay, you start seeing the pinching on either sides of those cells. When telophase is over, you got two identical cells. Okay, mitotic rate. It's just the number of cells you find at mitosis at any given time. Helps you estimate how frequently cell division is happening. So if you look at a slide, you're not going to see every slide in mitosis. Most of them are going to be in interphase because that's the majority of the time your cells are in a phase. So in general, the longer the life expectancy of a cell type, the slower its mitotic rate is going to be. Another way of looking at that is cells only got maybe a certain number of divisions in it. Okay, once it hits that number of divisions, it's pretty much done. It's kind of like your cell phone or other electronics where they say, you know, don't charge it up until it gets low because you get a certain number of good charges out of it. Okay? And stem cells go through mitosis very frequently. Okay? Because you have to replace a lot of more specialized cells. Okay? Those are stem cells. So they're going to go through mitosis more quickly than other more differentiated cells or the more adult cells. All right, so how is the cell cycle regulated? Well, I'm glad you asked. So cell division, again, is controlled by your genetics. Can be controlled by growth factors. These are factors, are chemical compounds outside your cells. They're extracellular. They can stimulate cell division, also growth. Tumors, or another term, neoplasm. Okay, these are masses. So this is like more of a medical application. So these basically abnormal cell division can cause the production of tumors. You get abnormal cell growth, abnormal cell division. Tumors can be either malignant or benign. Okay, typically the benign tumors don't threaten the person. And those cells tend to remain in what we call a connective tissue capsule. Whereas malignant tissues or tumors tend to spread from one part of the body to another, and they usually are more life-threatening. We also have a term called invasion. It's a process that happens when cancer cells spread to other tissues. Okay, so that being malignant, when they leave and go somewhere else, that's called invasion. Now, when those cells that are cancerous have moved somewhere else and set up a secondary tumor, then that's called metastasis. Okay, so the process of spreading is invasion. The process of setting up shop somewhere else is metastasis. Okay, the illness that's caused by those malignant cells is the cancer. Okay, so just make sure you understand the difference between all those terms. All right. Now, how does this process relate to who we are as adults? Well, we're all a combination of different types of cells that are differentiated. And what's differentiation? Well, it's basically specialization due to regulation of our genes, and it produces cells that have different capabilities that are limited. And specialized cells will form specialized tissues. And those tissues will have special roles. Okay? So it relates back to our levels of organization from chapter 1. Okay? So you have different kinds of cells, have different functions. Different structure, different function. Right? Remember, structure determines function. Okay. So what I'd like you to do is, well, before I do that, these terms, there's a lot of terms in this section. Basically, anytime you see these underlined words, be ready to see those in a matching. Okay, you could have a lot of these in a matching. Okay, and typically on a matching question, you're actually going to have more choices than there are actual spots. So you're going to have to really know those definitions. Okay, so there might be, 8 questions in a matching, but you might have 15 choices. Okay, so you got to know it's not purely just process of elimination. You really got to know your answers. All right, so what I'd like for you to do is to explain to your partner the phases of the cell cycle in mitosis. Okay, so I'm going to give you a minute to do that, and while you do that, I'm going to bring around tissue for you to wipe down your tables and chairs.